All right, so why don't we get started with the C-DEBI network series? Before we get started with the talk today, I just want to let people know that the next talk will be October 18th at the same time, 12:30 p.m. Rose is going to be talking about the microbial community composition and metabolic potential of a depth profile in the Tonga Trench from 400 meters in the water column to 2 meters below the seafloor. But today we're going to have Jackie has been nice enough to present a talk today for us. And just to give you a little background on Jackie, she received her bachelor's and master's degree at the University of Toronto, and she received her PhD from McGill University. And during that time, she went to the ends of the earth. She went from Antarctica to the Arctic, collecting samples and working on permafrost. More recently, she applied and received a C-DEBI postdoctoral fellowship, and now she's working at the Bigelow Laboratory for Ocean Sciences with Beth Orcutt. And she went to the ends of the earth once again to collect samples from North Pond, which she's going to be talking about today. So I've had the good fortune to be on at sea with her for about a year ago when she collected these samples. So looking forward to hearing about it, Jackie.
Great. Thanks, Jeff. I'm just going to turn off my video, and I hope that everyone can still hear me and see my screen. And maybe, Jeff, just give me a thumbs up if everything's okay still.
Yeah, it's good.
Great. Okay. Let me close this. Sorry, this platform is a little bit new. Okay, great. So first of all, I'd like to thank C-DEBI for giving me the opportunity to share some of my work, and especially for the postdoctoral fellowship that enabled me to come to Bigelow to work with Beth Orcutt, where I'm working on looking at microbial diversity and activity in oceanic crust environments using cell sorting techniques with the single cell sorting facility here at Bigelow. There we go. So first, I would just like to start off with a brief introduction to oceanic crust. For those who are not familiar with this particular subsurface environment, due to sheer volume, oceanic crust represents the largest aquifer on Earth, with large volumes of seawater entering into the crust through recharge sites like seamounts, then moving through the crust, gaining heat, interacting with the rock, changing in chemistry, before it discharges the crust through sites like hydrothermal vents. The volume of water involved in this is very large, with the entire volume of the ocean circulating through the crust on the order of every 10,000 to 100,000 years. So with the large volumes of seawater interacting with such large volumes of rock in the subsurface, you can of course expect that this particular environment is important for global chemical cycling due both to abiotic and biotic processes. Compared to even deep marine sediments, there's just a lot less known about the microbial communities in the crustal environment. What we know about the diversity, the microbial constituents, the biomass is based on relatively limited data sets, though these are generally low biomass environments, but nor is much known about the rates of microbial activity in oceanic crust environments. And these, this is all primarily due to the difficulties in accessing samples, especially pristine ones. Now, despite the generally low biomass of the environment, again, just due to the sheer volume, oceanic crust may actually represent a large proportion of all of the microbial biomass on Earth. So for folks that like to think in absolute numbers and on broad global scales, I'm showing a figure from Kara Haberling, 2010, which estimated the biomass that could exist within oceanic crust. Here on the y-axis is petagrams of carbon. Since then, others have made similar calculations and estimations, lowering the amount of biomass estimated to be in marine sediments. And a few days ago, Kara Magnabasco released a really wonderful review that also kind of lowered what we think the biomass in the continental subsurface may be. But the emerging picture from all of this is that oceanic crust is among the largest biospheres on Earth, at least rivaling marine sediments and the continental subsurface. And And importantly, there's evidence that this large biome hosts active microbial communities with large impacts on global biogeochemical cycling. For example, acting to remove large amounts of dissolved of DOC in the deep ocean. So this is clearly an important habitat to study, to understand global biogeochemical cycling. But because the crust is a low biomass environment, doing molecular analyses of the microorganisms inhabiting the rock, especially from bulk environmental samples, is really difficult. Extracting DNA is extremely challenging, and of course, even more challenging is extracting RNA from bulk samples in order to try to tie activity with genomics and phylogeny. And of course, another really big concern when you're working with such low biomass samples is the potential for incorporation of contaminants. So maybe from sample collection or processing, or the DNA extraction or sequencing. Here I'm showing a figure from Jessica Labonte from a recent paper from the Juan de Fuca Ridge, where they were able to extract and sequence the 16S RNA gene from 3 basalt samples and compared those sequences to sequence negative controls. And so these are the samples here in the purple. And here what you're seeing is everything that you see colored is a sequence that was found both in the sample but also in the negative control. And everything here in white represents a sequence that was unique to just that sample. The subsurface community is, of course, aware that contamination is an issue and a concern, and there are a number of approaches to dealing with this, including databases of common contaminants, and this is really, I'm saying this really just to highlight that to really get at the ecology of these environments, contamination is an issue that we have to be aware of and we have to contend with. So today I'm going to talk about work we're doing to try and overcome some of these low biomass limitations and caveats for molecular analyses. And I'm going to be talking about samples from 2 different subsurface sites along the Mid-Atlantic Ridge. The first using ultramafic rocks and sediments from the Atlantis Massif. This is a really interesting site where serpentinization occurs. And the other from a site called North Pond, where rock colonization experiments had been placed in observatories installed in the crust. And just briefly on serpentinization, for those who are unfamiliar, serpentinization is a geologic process that occurs when seawater interacts with rocks in the lower oceanic crust and upper mantle that contain minerals such as olivine, turning these rocks into serpentine. A byproduct of these reactions can be methane and hydrogen, as well as short-chain hydrocarbons and molecules like formate and acetate. And all of these are really great molecules to have as an energy or food source if you're microorganisms. So serpentinization has the potential to support life in the crust. Okay, and so how low is low biomass? Here are some cell counts generated by Yuki Morono on parallel samples from the Atlantis Massif from expedition 357. And they're really low, so ranging from 10 to the 4 cells to 10 to the 1 cells per cube centimeter. This is 10 to the 1 cells, really represents the limits of detection of this method, with the highest cell counts occurring in the sediments. And again, this is these biomass, these are really low, these are really difficult to extract DNA and RNA from. And so what we're trying to do is use flow cytometry to overcome these limitations. And our approach was to first try to separate and extract the cells from the environmental sample. We would wash and crush our rock, and then try to liberate the cells using a number of methods. Jessica Labonte tried using, for example, a Nicoden's density gradient. These are really great methods for cell counts, but are not really great methods for downstream genomic analyses. And ultimately what we found when trying to optimize these methods is that gentle is best. Getting the cells off of the rocks with just a simple vortexing and gentle sonication is really what worked the best, adding as little reagents to our sample as possible. Once we have separated our cells and resuspended them into a solution, we can tag them with a general stain, such as cyber green. And then we can use flow cytometry to sort them and concentrate them into a couple of microliters. So we're going from about 10 grams of sample to a couple microliters with 1,000 cells on average. So the idea is to get the cells concentrated into a small volume that we can then use for downstream genomic analyses. And we can do this both at the individual single cell level, or we can sort in consortia of cells. And so our approach to this was to first sort in consortia of cells to use amplicon sequencing to answer, which microorganisms are present, and then also to do mini metagenomes to ask, what is their metabolic potential, or what functional genes are there. And here I'm just going to very briefly describe the genomic analyses and what they tell us, since not everyone in C-DEBI, is a microbiologist and may not be familiar with these tools. Metagenomic sequencing involves the sequencing of the entire community at once. In our case, our community is about 1,000 sorted cells. And this results in, a lot of very short reads that end up having to be reassembled using computational tools. Once we've assembled our short reads into longer reads, we try to bin them into groups of fragments that we think all came from the same organism. This has been a really powerful approach to looking at microbial communities in recent years, but it does have some caveats. Sometimes our tools, for example, will take a fragment from one organism and mistakenly put it into another organism, as illustrated here with this purple fragment from this purple bacterium that's being, assembled into this red bacterium. But from here, we would annotate our genes, and the result is that you can make predictions about what the metabolic potential of the community is. And I want to emphasize here that having a gene doesn't necessarily mean that that organism is using that particular gene, or is even an active microorganism. It's, I think, better to think of it as a part list for the DNA that's present in your sample. Comparatively, single cell sequencing is more or less similar, except that you start off with a single organism. So all of your short reads come from a single organism, and that makes assembly a little bit easier. So you don't get those contamination issues. However, because you're working with such low amounts of DNA, or maybe that particular cell has a hard to lyse cell wall, for example, the completeness of the genome is likely to be a lot lower. And so our approach for these crustal samples was to do three different things. We were going, on our consortia of cells, our 1,000 sorted cells, we would first find out which microorganisms were present. Based on that information, we would choose for further sequencing, single cell sorting and sequencing, as well as metagenomic sequencing. All of these types of approaches require a whole genome amplification step, which is something that is known to bias your sample. It tends to selectively amplify certain organisms, but it's almost as a necessary evil when you're working with such low amounts of DNA. Okay, so first I'm showing some amplicon data from our Atlantis Massif samples. And in this slide, what I want to get at is this question of contamination. So here on the left, these are our samples that we were able to generate, greater than 30,000 reads after quality control. And on the right are the sequences, these failed runs. These are the sequences that usually, the sequence center would call failed. These generally had less than 600 sequences returned total. And here in the middle are our negative controls. And just as an overall first pass, you can see that our negative controls look a lot like our failed runs. Here in the blue is this, the dominance of a single OTU of Halomonas. Halomonas is a common seawater bacterium. It's also found in other crustal environments. And this is just to speak to the importance, again, of including these contamination controls, because Halomonas doesn't scream contaminant in this particular case. We took a pretty conservative approach to contamination removal, which was to remove all OTU that we observed in our negative controls out of our data set, resulting in the removal of about 160,000 sequences. Luckily, this actually didn't change the relative abundances that much. And there are two notes that I want to make here. One is I don't necessarily think that we have enough data here yet to say that the sorted cell approach is better at overcoming these contamination issues. This overcoming these contamination issues, presumably because we're not introducing the reagents of DNA extraction and things like that. But it is encouraging. But the second point I would like to make is I also don't want to make contamination control seem as simple as just subtracting all of your OTU and your negative controls. There are a number of publications that show you can really bias your data doing this. There is no one fit all. And I'd be happy to talk with anyone a little bit more about my thoughts on this afterwards if anyone wants to hear it. But now I would like to highlight some of what we're seeing in these samples. The letters that you see on the top, the C and S's, indicate whether it was a crustal sample or a sediment sample. Generally, even though we had fewer sediment samples to work with, the ones that were successfully sequenced tended to be sediment samples compared with our crustal samples. Here in the blue, these gamma proteobacteria that you see also all belong to Hilomonas. This is a different OTU than what we saw in our negative control. We also see an acetobacteria dominating one of these samples. This was similar to those found in other serpentinite-hosted springs in the tablelands. Here, these two crustal samples were dominated by a mesorhizobium. Mesorhizobium have been found in oceanic crust and the terrestrial subsurface, but have also been found in databases of common contaminants. Again, speaking to the care that we need to take. Here in the black, these are sequences that could not be identified to the phylum level, so we have some potential novelty within our sequences. And here in the red, in sample 69A, 4R1, as well as 70C, 3R1, we see that our samples were dominated by candidate phylum Data bacteria, formerly known as SBR 1093. This particular organism is interesting because it's been found in oceanic crust and seafloor rocks and ferromanganese nodules before. But really interestingly, this particular organism is a putative autotroph using the hydroxypropionate hydroxybutyrate cycle, a cycle previously only known in archaea. So based on these data, as well as just the general, higher success rate with sediments we were having, we chose this particular sample for single cell sorting. And for the metagenomic sequencing, we chose 2 sediment and 2 crustal samples. So first I wanted to talk about the single cell sorting. We sorted 384 cells. 94 of these could be identified with 16S rRNA sequencing. This is using, old-fashioned Sanger sequencing on the amplified DNA. So you can kind of think of the scale a little bit like a clone library. And here, that's what this seg plate down here is. And you can see that the distribution and the diversity of these sorted cells is very different than what we were getting in our amplicon sequencing. This may be an artifact of using the MDA. We had a lot more diversity. We were only able to identify one single seg of Data bacteria, but we were able to identify several other segs that were interesting, including candidate phyla Scheckman bacteria. This is a putative sulfate-reducing carbon monoxide oxidizing organism, as well as the CM66 group of chloroflexi, for which basically nothing is known other than where we can detect it in amplicon sequencing. And I had hoped I would get these genome data back by the time for this talk, but I actually only got it on Tuesday. So hopefully you'll see me give another talk soon. And we have 230 single amplified genomes to work with, and I'm very eager to see what's within them. But in the meantime, we can turn to metagenomes, the megs. From our metagenomes, I was able to assemble 33 megs. Generally, most of them had less than 50% completion, but they had, I would say, tolerable amounts of estimated contamination. And here what I'm showing is just the presence or absence of some key genes in general metabolic pathways in either the crust or the sediment metagenome. And again, because this is a site with potential hydrogen production and methane, I was really interested in looking at hydrogenases or any of the key genes involved in methane cycling, but I couldn't find any. In terms of potential autotrophic pathways, the only key genes I could find was the carbon monoxide dehydrogenase acetyl-CoA synthase complex. I found this in one meg that was recruited from the crust entirely. This protein is part of the reductive acetyl-CoA pathway, which allows for assimilation of carbon monoxide. However, in other megs recruited from both the sediment and the crust, I could find copies of carbon monoxide dehydrogenase, just not an assimilatory carbon monoxide dehydrogenase. So they were in a total of 4 different organisms. 3 of them were acetobacteria. There is, to date, no experimental evidence of carbon monoxide assimilation by an acetobacteria, and these are, I couldn't find any other genes associated with carbon fixation in these megs. These are likely using carbon monoxide to supplement their energy rather than a carbon source. And in the SAG data that I got back on Tuesday, we have about 21 of these organisms that I will be able to probe in the future. I also had a D-halococoidetes meg that was only recruited from the crust. It had the assimilatory CODH, as well as other genes in the Wood-Longdal pathway, but not a complete pathway. This particular meg also had some denitrifying genes, so this is potentially a carbon monoxide oxidizing denitrifier. In the case of the D-halococoides, we have 24 SAGs that we can probe. And because, that Dada bacteria was really the reason I championed this particular sample for single cell sequencing, just as a follow-up, I could find absolutely no genes associated with autotrophy. There's every indication that this is actually an aerobic heterotroph. But just to sum up what I've talked about so far really briefly, so using these methods, using flow cytometry, we were able to sort cells to successfully get amplicon sequencing, metagenomes, as well as single cell genomes, which are now in hand. But I don't really want to downplay, how difficult this has been. So, out of about 51 environmental samples, after sorting, about 29 of them, 30 of them were able to get amplifiable DNA out of, and a little over half of those we were able to successfully sequence. So yes, this is working for concentrating low biomass samples, but it still by no means doesn't make low biomass samples easy. Also up until now, I've been exclusively talking about getting at genomic data, but this doesn't really tell us anything about the activity of the microorganism or the state of the cell in the environment. So is it viable, is it active, or is it dormant? These cell states are very important to understanding the ecology in any environment, but especially in some of the low energy environments that the C-DEBI community tends to study. And so to get at this question of activity, we've been using a method called BONCAT. What we would do is we would incubate our environmental sample with a fake amino acid called HPG. This is an analog to methionine. Any organisms that were actively making and synthesizing new proteins should incorporate this fake amino acid into their new proteins. There's every indication that this works on a broad range of microorganisms and is seemingly independent of metabolism. So we're not limited to identifying the activity of one particular microorganism. From here, you can do a click chemistry, which causes those proteins which have incorporated this fake amino acid to fluoresce. resulting in cells that are fluorescent. And from here, you can visualize via microscopy, or you can do flow cytometry and do all of the genomic analyses that I just described. And so we did these on the rock chips that were incubated in the North Pond subsurface, and we found that, yes, we can detect active microorganisms. So, here on the left, I'm showing, or here on the right, I'm showing some before and after pictures of sterile rock chips, and then the after of when they came out 6 years later. Here's some SEM photography that shows that, yes, these rocks were colonized by microorganisms. There seems to be some secondary mineralization happening. But here in the green, these are fluorescent cells that have incorporated this fake amino acid from our rock chips. And so we did single cell, or we did consortia sorting as before, where we would sort 1,000 cell sorts on both BONCAT sorted cells, here indicated by active, and then also on fiber sorted cells, which would sort the total community. And, briefly, for some samples, like this B10 basalt, we see that the differences between the active community and the total community are quite different. But in other cases, the dominance of the communities present and the active communities, like here in the olivine, with this dominant presence of Halomonas with organisms that seems to be everywhere, they seem to be quite the same. Here in our total communities, we also see Dada bacteria pop up again, but only in our total communities, not in our active communities. So the idea here is to separate active organisms as a way of getting better targets for our omic analyses, to sequence, to better understand what's going on. And so the ultimate goal is to get BONCAT down to the single cell level. We've sorted at the single cell level, but unfortunately our sequence data has not come back. So I don't know if it's been successful. But I thought I would pause on saying, why down to the single cell? And I really think that, while we have metagenomic binning techniques, they're not always perfect, they are powerful, but I think what SEGs really allow us to do is get a high resolution look at genome characteristics and strain variation that you really just can't get when you assemble multiple organisms together in MEGs. My particular interests lie in looking at molecular adaptations to these low energy environments. And because many of these organisms also occur in energy rich environments, such as a Halomonas, there's an opportunity here for us to use comparative metagenomics to uncover, what some of these adaptations may be in environmental organisms without the need for culturing them. Comparative genomics, is a very powerful tool for this, but it's largely being reliant on cultured isolates, which are the minority of the organisms out there. So getting this type of resolution required for this is very difficult with MEGs, but single cell sorting really does enable this, and targeting active single cells in particular, I think, has a potential for us to really narrow the scope of the organisms that we do these analyses on. So just as a quick summary, low biomass samples are difficult to work with. A lot of people in the community know this, but BONCAT, I think, can identify microorganisms at the single cell level, and has a really great potential for us understanding the ecology and the molecular adaptations of some of these low energy environments. And so with that, I would just like to acknowledge a lot of people, Beth Orcutt and all of the members of the Orcutt lab, the Single Cell Genomics Center at Bigelow, especially Nicole Polton, who has done a lot of the optimization with me from the Single Cell Genomics Center, and Jessica Labonte, who did a lot of the method development for trying to get the cells off of the rocks for downstream genomic analysis. And with that, I will see if I can share my video and take any questions.
Hey, thank you very much. That was great.
And okay, found the chat box. Perfect. Okay, so Jesse asks, great data. Approximately what proportion of your rock chip cells were active or incorporated BONCAT, and over what incubation time? Did you perform any time series incubations to get a sense of the fraction of cells that had different rates of transcription? So no, I did not perform any time series incubations using BONCAT to get, this would get at active rates of translation, not transcription. However, I did do time series incubations with a nucleic acid analog. So these are all amino acid analogs to look at translation, and I've done parallel experiments with nucleic acid analog to try to get rates of replication. I don't have those cell counts, but I know that you're interested, and I will let you know how those go. And in terms of what proportion of your rock chips were active versus incorporated, so the incubation time was 24 hours on ship, and active versus incorporated. I can't answer this based on this data. Using microscopy, I only looked at the active proportion, and when I do the sorting, I do the active. It's not quantitative, I guess, is what I'm trying to get at. I can't quantitate it. Yeah. Do you couple BONCAT to fish at all? No, I don't. No, we're just coupling the BONCAT to to the genomic analyses. And then from Roland, if I understood correctly, your total community was bulk data. BONCAT was 16S of 1,000 sorted cells. If that is correct, have you considered treating all data compositionally? You are randomly subsampling all cells. I'm not, Roland, can you maybe elaborate a little bit more on what you mean by treating all data compositionally? I don't know what you mean by compositional data analysis. Do you mean just like the overall composition of the community? We are randomly subsampling the cells. Oh, like the presence of a cell may be due to another dying. You mean after, because your analysis is limited because you're looking at 1,000 cells. So the reason between 1,000, the reason, maybe I'll give some context for the 1,000 cells. Basically, for all of our samples, I would try to sort as many cells as possible. Often we didn't even make the 1,000 cell mark. They're just so low biomass. If we did hit the 1,000 cell mark, that would be kind of the stopping number to say like, okay, we can actually try and get a replicate of this. And so we have very few samples where we do actually have successful replicates. And the replicates have different relative abundance, but the composition of the microorganisms are there. And so when you so Roland further asks, so when you analyze those cells, did you MDA prior to PCR or just a lot of PCR samples? So all of the for the amplicon sequences, we do MDA and then send the MDA DNA out for, yeah. So when we do these and that, so when we do the 1,000 cell sorts, I MDA it in the lab, and then I send that DNA out for sequencing. And I do a PCR check in the lab just to see if I actually, get a band or not, prior to spending a lot of money on the sequencing. So that's what the PCR is for. It's just an in-lab check to see if we're going to send it out for sequencing. But other than that, the only PCR that actually gets done on our samples is for the Sanger sequencing. Does that answer your question, Roland? Great. Ooh, this is a great question. So Alvaro says, you have 3 different ways to describe the community here. Which one would you say reflects better the in situ microbes of your samples? That is a great question that I couldn't say with a lot of certainty. Each method here has its own bias. Certainly, you know, even our starting step of extracting the cells will have a bias. You know, for example, maybe we're using very gentle vortexing and sonication to shake the cells off of the rocks. So, you know, maybe we're already biasing it to be kind of the cells that don't hang on as strong. So there's a lot of biases. I really think, based on the ones that we have extracted, my hunch is that the single cell sorting reflects better the diversity, especially in the relative abundance, because we're not relying on the MDA, which is that necessary evil, but because we're not relying on MDA to get a sense of what is in our community. And what were the incubation conditions with the analog? Those were at 4 degrees Celsius.
Well, if we don't have any other questions, I want to thank Jackie for giving this talk. Any other questions? I guess that's it.
All right. See you later.
All right. Hey, thank you very much.
Yeah. Thanks, Jeff. Bye.
All right.